Good morning. This is part 4 of heredity, and before discussion of the new concepts in part 4, we're going to highlight certain older concepts that are a bit confusing. So we talked about a random distribution of chromosomes around the equator of the cell in metaphase of meiosis 1. And we also talked about independent assortment of alleles in metaphase 1 for the free alleles, meaning that here we are talking about, to the left here, we are talking about random distribution of chromosomes, and to the right, we are talking about independent assortment of alleles if they are free alleles. In random distribution of chromosomes, this simply means that if we consider that the red chromosomes are the maternal ones and the blue chromosomes are the paternal ones, then we have two possible arrangements. In that first arrangement, you have, if you are formed from this gamete to the left, then you're going to take the dominant allele A and the dominant allele B, and accidentally you also took the dominant alleles D and C from your dad. But if you're formed from the gamete to the right, then you're going to take the recessive alleles from your dad, A small, B small, and the recessive ones from your mom. This is just an example for a possible arrangement of the chromosomes around the equator of the cell. But there is another possible arrangement like this. In such a case, if you are formed from the gamete to the left, you will be formed from maternal chromosomes only, and you will receive two dominant alleles for A and B and two recessive alleles for C and D. Okay, so here this makes the difference. The independent assortment, the two possible arrangement of the chromosomes around the equators made that the possibly produced gametes have different chromosomes with different alleles carried on them. So another thing is that not only does random distribution of chromosome results in variation of the genotype of the produced gametes, but in the first case, when you were formed from the gamete to the left, you were formed from both paternal and maternal chromosomes. But in the second arrangement, you took both maternal chromosomes. So you might be saying, or asking yourself, didn't we say that random distribution of chromosomes ensures that we take both paternal and maternal chromosomes? Yes, if we are considering the whole cell, if we are considering the whole set of gametes, then definitely, because of the different possibilities of arrangement of the homologous pairs around the equator, then most probably every gamete will be a blend of both maternal and paternal chromosome. Which is predominating? Do we have more maternal than paternal, or do we have more paternal than maternal? This does not matter, and meanwhile we cannot expect it because it occurs at random. But in the possibility, the lowermost possibility, which is this one, it happened that you received two maternal chromosomes in one gamete, but this is because we are considering only a small number of chromosomes. So what if we are talking about independent assortment of alleles? Now, if we consider the same independent assortment of chromosomes that we discussed on the left half of the screen, but now we are considering the alleles on them. Now, when A and B. Let's consider, for example, that allele which is A capital and that allele which is C capital. A capital and C capital are present on, they are alleles for two different characteristics, and they are present on two different chromosomes, and therefore they are considered free alleles or non-linked alleles. That's why when they exchanged their positions, if you try to get the difference between the uppermost cell and the lowermost cell, you will find that this chromosome to the left, that one has passed to the right. However, this one stayed in its place. So A capital is now in the right half, and C capital is now still in the left half. And this indicates that because these two alleles are independent, as there is random distribution of the chromosomes on the gametes, there is also random distribution of the alleles that are carried on them, and this is only allowed because the alleles are free. But if we consider alleles that are linked, like A capital on that chromosome and B capital, they have to go together. If the chromosome is to the left, then A and B will be to the left. If the chromosome goes to the right, A and B will stay together. But this means that they are not separated from each other unless crossing over occurs. But this doesn't mean that there is no variation in the genotype, and this is very important, and this is where the confusion is. So although A and B are linked, and although they go together to one gamete, however, they might be in the gamete to the left, or they might be in the gamete to the right. So again, random distribution of chromosomes around the equator can not only result in random assortment and distribution of the free alleles on the gametes, but it can also result in random distribution of linked alleles, but in a very restricted sense, which is which gamete are we going to go together to, meaning that A and B can be on the right side or on the left side, but they go together. You might also be asking yourself, does it matter if, or does it make any difference if A capital and B capital are to the left or to the right? If they were alone, it wouldn't make any difference if they were alone in the cell, but they are with other chromosomes. So here, for example, when they were to the left, okay, they were with C capital and D capital. But here, when they went to the right, they are now with C small and D small. So random distribution of chromosomes in metaphase 1, if meiosis 1 proceeds, then we are going to go until telophase 1 and cytokinesis, and therefore those chromosomes will also be randomly distributed on the gametes, and we will have variable gametes. Similarly, independent assortment of free alleles in metaphase 1 or the restricted independent assortment of the linked alleles results in random distribution of those alleles on the gametes and also result in variation. If we add to this the crossing over that might have occurred in prophase 1, so we are plus, of course, the random fertilization, then we are gathering like 4 causes of variation. Now, again, for the question we had in the homework, we emphasize that random distribution of the chromosomes on the gametes, ensures that each daughter cell receives both paternal and maternal chromosomes, especially if we are considering the whole number of chromosomes. And the random distribution of alleles on the gametes ensures that the daughter cell also contains both paternal and maternal, but this time alleles. So you have to be very cautious when you are using the terms. Are you talking about chromosomes or are you talking about alleles? Now, the new concept we are going to discuss is the chromosomal map. And actually, the diagram you see here on the slide is, or on the screen, is a chromosomal map. The chromosomal map is a diagram, and that diagram represents the position of the alleles on the chromosomes. Actually, it represents the relative positions of the alleles on the chromosomes. And when I say relative positions of the alleles on the chromosomes, I mean where are they located in relation to each other. So here, for example, we can say that B lies in between A and C. Or we can say that B is close, and we can say that B is closer to C than it is closer to A. Chromosomal maps, how can we draw the chromosomal maps? There should be a basis that determines the distance between the alleles on the chromosomal maps. This is based on the results of genetic experiments. Which kind of genetic experiments? In which inheritance cases? Whenever we have linkage and crossing over. As we saw before, when we did a dihybrid test cross, we had, for example, 33 parental individuals, and then we had 17 recombinant individuals with a recessive and dominant, and then 17 with dominant and recessive, and then we had 33 again, meaning that we had a parental ratio and we had recombinant individuals. And as we said before, in this dihybrid test cross, there is no fixed result. The result depends on how frequent crossing over occurs. And frequency of crossing over depends upon the distance between the two alleles on the chromosomes, meaning that the bigger the distance between the two alleles on a particular chromosome, the higher the possibility or the likelihood of crossing over to occur between the alleles. And this sounds logical, because if the two alleles, for example, A and B, A capital, B capital, and A small, B small, are very close on the chromosome, there wouldn't be place for crossing over to occur. For example, if this is the first chromosome and this one is its homologue, then if I have A small here and B small here and E small here and F here, this small difference would not allow crossing over to occur. I'll pause for a second and then resume recording. Hello again. So we were talking about chromosomal maps and saying that the distance between the two alleles on the diagram of the chromosomal map is an indication of how far the two genes are located or are away from each other on the same chromosome, and that the greater the distance, the greater the likelihood of crossing over between the two alleles. For example, in that diagram on the screen, you can see that the distance between A and C is bigger than the distance between A and B. Therefore, the likelihood of crossing over between A and C is greater than the likelihood of the crossing over process to occur between B and C. Or between, so again, I'm sorry, I was distracted. The likelihood of occurrence of crossing over between A and C is greater than the likelihood of crossing over between A and B or between B and C. Now, if we consider the distances that are written on the diagram, then we have the distance between A and C is 18 units, and the distance between B and C is 6 units. These units are arbitrary units, meaning that they are conventional units. Every unit of these or this unit is called a map unit, or it is called a centimorgan unit. And centimorgan unit, the unit, only one unit, say for example, we are dividing the 6 units into 6 parts, then every unit of these is called a centimorgan. And a centimorgan represents the possibility of 1% of occurrence of crossing over between two alleles. So if the two alleles have a distance of 6 map units or 6 centimorgans between them, this simply indicates that there is a 6% frequency of occurring of crossing over between B and C. And if the distance between A and C is 18 units, this means that there is an 18% frequency of occurrence of crossing over between those two linked alleles that are present on the same chromosome. Of course, as we have just mentioned, the frequency of occurrence of crossing over between A and C is 18%, and it is bigger than that between B and C, which is 6%. This simply occurs because of the greater or smaller distance between the two alleles. Now, coming back to the frequency of crossing over, if we visualize a mother cell or a parent cell or a germ cell during prophase of, during metaphase of meiosis 1, meaning that it is visualized during metaphase 1. So we have the two sister chromatids of the paternal chromosome and the two sister chromatids of the maternal chromosome. So synapsis has occurred, and we can visualize a tetrad. I've selected dominant alleles for the paternal chromosome and totally recessive alleles for the maternal chromosome, just for one simple reason: to make the discrimination or the differentiation easier. And of course, as you can see, the paternal chromosome is duplicated and the maternal chromosome is duplicated because we are still in metaphase 1, not in metaphase 2. If we assume, for example, that I'm assuming that the distance between alleles A and B is 17 units, this means that on the paternal chromosome, the distance will be 17 units, and on the maternal chromosome, although the alleles are recessive, the distance is also 17 units. And if we say this simply implies that the frequency of crossing over is 17%. What do we mean by the frequency of crossing over is 17%? We said that every unit represents 1%, or every unit represents a likelihood or a probability of 0.01. So if they are 17 units, then the frequency of crossing over is 17%. What does this mean when we say that the likelihood of occurrence of crossing over between A and B is 17%? This means that out of every 100 mother cells, or germ cells, 17 will show crossing over. How does crossing over look like? How does it occur? And how does the cell where crossing over occurs in prophase 1 and therefore is visualized in metaphase 1? We will see that there will be exchange of homologous parts of the chromosomes. So the maternal B small will go to the left and the paternal B capital will go to the right. And then after 2 turns of meiosis, meaning that the 4 daughter cells you see here in that line occur after 2 rounds of meiosis, we imagine that meiosis 1 has occurred and that meiosis 2 has occurred. A capital, B capital will go to 1 daughter cell. On the other hand, A small, B small will go to another daughter cell or to a second daughter cell, and both of them are non-recombinant. The first one is called a parental 1 gamete because it contains a parental 1. We're focusing on 1 chromosome as if the cell was containing 1 chromosome. So we have parental, a parental 1 gamete that is similar to the first paternal chromosome. And then we have a parental 2 gamete that is similar to the second maternal chromosome. And in between them, we have recombinant gametes that contain recombinant chromosomes, apart from the paternal and apart from the maternal. And since they contain recombinant chromosomes, we call them recombinant gametes. If 17 cells out of 100 cells carry out that process, we will find that every daughter cell we have here, we are considering 1 daughter cell, will produce 2 recombinant gametes. And therefore, the rate of recombinant gametes or the recombination frequency is 34%, and it is double the frequency of crossing over. And this is very important. The frequency of crossing over is the percentage of mother cells or parent cells or germ cells that carry out or that show crossing over. And this simply indicates you that not all gametes will undergo crossing over. Sorry, not all mother cells will show crossing over, but for what? For this particular pair of chromosomes. Of course, at the end of the day, we will find that all germ cells will show some degree of crossing over in between some homologous chromosomes, okay? But here, why are we saying that out of every 100 cells, only 17 show crossing over? Show crossing over for that particular pair of chromosomes or that particular tetrad that you are seeing. So if they show if 17% show crossing over for that tetrad, maybe for another tetrad, 20% would show crossing over. And every cell for that pair of chromosome produces 2 recombinant chromosomes. So the 17% would produce 34% of the gametes that are recombinant. So the recombinant frequency is 34 out of 100. Now, what about the ratio? The ratio is, now these are the 34, but we divide them into 2 because they have 2 different genotypes. Here we have A capital, B small, here we have A small, B capital. That's why we split them into 17 and 17. And then we have 33 and 33, which is the remainder of the 100. 64 minus 100 minus 34 makes what? 66 divided by 2. Then we have 33 and 33. Okay? So the chromosomal maps are a graphical representation or a diagrammatic representation of the positions of the genes, or actually, more correctly speaking, of the alleles on the chromosomes that are obtained by observation of the chromosomal bands. Of course, if you are visualizing it under the microscope, or because there would be, if you stain the chromosome, you would have bands of different staining and different shades of gray and black and white. or by determining the degree of linkage and crossing over between the genes, and this is done through genetic experiments. And then we talked about the centimorgan or the map unit, and we said that the centimorgan or the map unit is a unit for measuring genetic linkage, and it is defined as the distance between two loci. So when you say gene, you can say gene loci or simply between two alleles in which the expected average number of chromosomal crossing over is 1%. This is the definition of the map unit. The frequency of crossing over is how many crossing overs will occur per generation between two specific genes. Gives no information about the actual distance between the genes. We don't know the actual distance between the genes since we are not geneticists and we're not working in genetic labs, but we can just know the relative position of the alleles. So we can know how far the genes are. The farther they are apart from each other, the higher the frequency of crossing over. And we can also know the order or the arrangement. So you know two things from the chromosomal map: how two particular alleles are far from each other on the chromosome, and what is their order or what is their arrangement. And we're going to see an example. So if we look here at the chromosomal map, we have plenty of information that we can say, but I'll pause for a second to have my coffee and then resume. When you get chromosomal maps on a test, for example, you might have two different shapes of the chromosomal map. It might look like this in the form of straight lines, or it might be a 3D diagram like the one below. If we consider the one above here, we have 4 alleles, B, A, D, and C. They're giving you the percentage of the frequency of crossing over between B and D. Okay, and this simply means that the distance between B and D is 40 units, and they're giving you the frequency of crossing over between A and D, which is 25, and therefore the distance between them is 25 units. And then we have the frequency of crossing over between A and C, which is 30%, meaning that the distance between them would be 30 units, and that between B and C would be 45 units. So where would be the question? The question might, the question might be here. For example, they might ask you about the frequency of crossing over between D and C. In such a case, you would say A and C is 30 units minus A and AD, which is 25. Then here you have 5 centimorgans or map units, and this indicates that the frequency of crossing over is 5%. On the other hand, if we have the distance between B and D being 40 units, and that between A and D is 25 units, then the distance here would be 15 units, which is the difference between 40 and 25, and this simply indicates you that the frequency of crossing over is 15 between B and A, because we have just said that the number of crossing over in a single generation is 1% for every unit, okay? Of course, the linkage map can be read like this, BADC or CDAB, if you want to read the relative positions between the alleles. Similarly, here we have, you have the frequencies, and according to those frequencies, we are going to determine the distance. So here you have 17%, the total percentage or frequency between body color and wing size. And then you might be given that one, for example, which is 8%, and then the remainder would be 9% or 9 units. Now, here we have the results of a genetic experiment. Here, as we did before, we have 2 recombinant gametes and 2 non-recombinant gametes. So because here we have recombinant chromosomes, then they would result in recombinant gametes. Here we have non-recombinant chromosomes, and therefore these 2 gametes, if those were recombinant gametes, those are recombinant gametes, then those are non-recombinant gametes, or you just say parental gametes, whether one of them would be resembling the first parent and the other resembling the second parent, okay? And if we cross them with a recessive individual, you know, of course, that crossing over in recessive individuals makes no difference. So if I exchange, for example, this B with that B, I will still obtain the same results. So crossing over in recessive individuals does not result in variation. And this is very important. So crossing over in recessive individuals results in no variation. So here, if we have those blue chromosomes are coming from a recessive individual, so we have a constant genetic makeup of the chromosome, and here we have 4 different possibilities. In those 4 different possibilities, we have, of course, notice that if you have a certain ratio, the number of individuals is not sharply determining the ratio. So, for example, here we are supposed to have 2 different parental genotypes. This is parent 1 and this is parent 2, but they are not exactly the same number, but they're close to each other. And here, so one of the parents was definitely having a gray body and a normal length of the wings. The other one was purely recessive and has a black body and vestigial wings. But when we did the dihybrid test cross, as we mentioned in part 3, we had 206 individuals that are taking the dominant color from one parent and taking, which is dominant, the gray color, and taking vestigial wings from another parent, so they are recombinant, and we have 185% that are recombinant. So if we are calculating the recombination frequency, we are not calculating the frequency of crossing over, but we are calculating the recombination frequency. So we have, for example, you're going to sum up or to add up 206 and 185, and this will give you 391 recombinant individuals. You divide them by the total number of offspring. So in the denominator, you're going to sum up 965, 944, 206, and 185, and then multiply by 100. And you will find that the recombination frequency is 17%. Okay. Now, this was the first non-Mendelian condition, which is complete linkage, because, as you know, Mendel studied only 3 alleles that are present on separate chromosomes. The second non-Mendelian case, or the second inheritance case that does not follow Mendel rules, is lack of dominance. And in such a case, we would find that the characteristic, like the color of the fur in certain species of mice, for example, is notice that animals like mice have different types of inheritance of the fur color. So in some species, for example, the black dominates totally over the white. But in other species, there is lack of dominance, and the black and the white merge together and give a gray color, which is a condition of lack of dominance. So you should like answer the genetic problem or solve the genetic problem according to the information that is given in the problem and not according to previous knowledge. So in case of lack of dominance, we will have two homologous chromosomes, and on each homologous chromosome, we will have an allele. For example, if the maternal allele carries the A chromosome that is responsible for the white color of the mouse fur, and B capital is responsible for black, then at the end of the day, you will find that the mouse will be gray, meaning that the phenotype of this individual, we don't call it hybrid individual when we are not talking about a Mendelian case. The phenotype of that individual that carries the different alleles is a blend of the two original alleles. So it's a blend, meaning that we can also express it as being intermediate between white and black. So whenever you see, for example, two parents, a white furred parent and a black furred parent that get a gray furred mouse or offspring, then most probably you're dealing with a case of lack of dominance. In lack of dominance, neither of the two alleles is dominant. Neither of the two alleles masks completely the other allele. So there is no dominant and recessive, but both are expressed on the phenotype, and not only are they expressed on the phenotype, but they blend together. Most probably the A allele is expressed or is translated into an enzyme, let's call it an enzyme A, that forms a certain pigment, and B forms another is translated, B allele is translated into enzyme B, and every enzyme activates a particular chemical pathway, and at the end of that chemical pathway, we obtain a pigment. So when both pigments are produced in the cell, here, for example, we have a white color, so we are not sure whether it's forming a white pigment or not forming a pigment at all. But say we have, for example, black and yellow, as in some types of guinea pigs, for example, then in such a case, enzyme A will activate a pathway, which we would call a pathway A, and pathway A would result in the formation of a pigment A. And at the same time, we would have enzyme B that, when produced in the cell, activates a pathway B that results in pigment B. In lack of dominance, the pigments blend together. We have several examples, like, for example, here, these are the parents, a black parent and a white parent, and they result, the F1 generation will all be B capital, W capital, which is of gray color. Why do we write the two alleles in capital letters? Here, because there is no other choice for us. You cannot put B capital W, for example. It cannot be as in heterozygous dominant Mendelian cases, because if you have BW, it would be gray. So if the mouse is black, then definitely it is B capital, B capital. And if that mouse is white, then it is definitely WW. Okay? Here, similarly, if we have a long watermelon with round watermelon, then we would have all the first offspring, BR capital, L capital, which are oval. The third example is the Japanese 4 o'clock flowers. So if we have a red flower and a white flower, then the first generation will all be pink flowers, which is intermediate between the red color and the white color. Okay. Now, in lack of dominance, we on the previous slide, we just considered first generation. But if we take, for example, 2 pink flowers of the first generation, and now they are the parents, so we call them parent 2, not parent 1, and we cross them with each other, then we would have, for each of them, we would have 2 possible gametes, RW and RW. And if we do the proper genetic diagram, first with first gives R capital, first with second gives RW, second with first gives again RW, and then second with second will give you WW. So here we have 25% of the offspring be red, and 25% of the offspring being white, and we have the pink offspring. So the ratio is actually 1 to 2 to 1, 1 parental 1 parental 2 to 2 intermediate, or a blend between the two. Do we need test cross in case of lack of dominance? No, we do not need a test cross. Why? Because when is test cross needed? In complete dominance. If the condition was a case of complete dominance, then if you have a red flower, and this is how we write a hybrid individual, for example, a hybrid red or pink flower in pea plant, this is the dominant allele, that is the recessive allele. The dominant allele completely masks the recessive allele, and the flower is red. Similarly, if it is R capital, then it is red. So here, for a red flower, we have 2 possible, for a red flower, and red is a phenotype, we have 2 possible genotypes. Either it is pure or homozygous, or it is hybrid or heterozygous. So in order to differentiate between the two, you need a test cross. But here, in case of lack of dominance, you don't need a test cross, because if you have 2 different alleles, this will be apparent on the phenotype, and it would be the intermediate phenotype, or the blend between the 2 parental phenotypes. The third non-Mendelian case we are going to talk about is the condition of codominance. And in such a case, in codominance, and the best example is that of the blood groups, in codominance, you have 2 different alleles. And when the 2 alleles are, if each allele is present in its homozygous form alone, then there would be no problem. But the problem is when those 2 different alleles are present together, what would be their effect on the phenotype? For example, in the regular blood grouping system, which is the ABO system of blood group, which is the most dangerous, because as you know, in the plasma, we might have antibodies, and these antibodies may cause agglutination of the red blood cells that contain the same antigens. For example, if you are blood group B and you have in your plasma anti-B, and if you receive a red blood corpuscle carrying antigen B, then you will attack it, resulting in either agglutination and sticking of those red blood corpuscles and clogging of the small capillaries and death of the organs, or this might result in hemolysis and rupture of the red blood corpuscles, release of all the blood, of all the hemoglobin that is inside them, and a condition of hemoglobinuria, fever, and shivering of the patient, and the patient would be in a very miserable condition. The ABO blood system depends on the presence of three alleles, or three different genes. So we have allele A, allele B, and allele O. But actually, on the specific chromosome that is supposed to carry the alleles for blood grouping, you have only two loci. So you only receive two out of those three alleles. So first of all, inheritance of the ABO blood system is considered, as we said before, a condition of multiple alleles. So this is the first breach of Mendel's law, that we have three alleles controlling one phenotype, which is the blood group, and that's why we call it a multiple allele. The second problem is that we know that A is dominant over O and B is dominant over O. O is recessive to both, as we said before, and that will be repeated later on. But when it comes to AB, suppose your genotype is AB. So your red blood corpuscle will express on its cell membrane, because you have, those are genes, those are alleles. So this is your genotype, but how will this be expressed on your phenotype? Your phenotype here will be the antigen that will be present on the red cell membrane. So you will express both glycoproteins or both antigens on your red cell membrane, meaning that your red cell membrane will contain A and B. So what would be your blood group? Your blood group would be AB. So in such a case, neither of the two alleles is dominant over the other. So there is no complete dominance as in Mendel. Furthermore, there is no blending between the two, meaning that A has its separate identity and B has its separate identity because they are equally potent. And in such a case, we call this condition a condition of codominance, okay? So up till now, blood grouping has shown codominance and has shown multiple alleles. What's the evidence that they do not blend and that they are equally potent? That the evidence is that they act separately in the blood, meaning that if you have anti-A in your plasma, it will attack the red blood cell, and if you have anti-B, it will attack the red blood cell. But anti-A attacks the red blood cell through attacking antigen A, and anti-B attacks the red blood cell through attacking antigen B. So they have separate entities. The same applies to the other system of blood grouping, which is the MN blood group system, the MN blood group system. But the MN blood group system is less important and less serious than the ABO blood system, and we don't take it that much into consideration when we are considering blood transfusion between the patients. Why? Because the antibodies against the M antigen and the N antigen, I mean anti-M and anti-N antibodies, do not react at the normal body temperature. So if you have antigen M on your red blood corpuscle and you receive plasma containing anti-M, there is no risk of antigen-antibody reaction. There is no risk of antigen-antibody reaction, and therefore no risk of agglutination or hemolysis. Why? Because they do not interact at the normal body temperature. Now, what are your possible genotypes? You might, of course, there are still two loci, just like in the ABO blood system. So you might receive M and M, or you might receive M and N, or you might receive N and N. If you receive M and M alleles, there is no problem, because your red blood corpuscle will express M on your RBC. If you on the other hand, if your genotype is NN, there is no problem, because you will have N antigen on your red blood corpuscle. But if you receive allele M on one chromosome and allele N on another chromosome, then you will have both on your red blood corpuscles. So allele M is expressed as antigen M, and allele N is expressed as antigen N. This is important because you're always confused about this. So we have an allele, which is a gene, or the one of the alternatives of the gene, and it is expressed as an antigen on the surface of your red blood corpuscle. Here, the allele represents your genotype, and it is present in the nucleus, but the antigen represents your phenotype, and it is present on the RBC membrane, or on the red blood corpuscle membrane. Okay, this was the third non-Mendelian case, and here we can write the genotypes in a different way. So if your phenotype is M, then you definitely inherited M and M, and you write them on an L capital. So you have L capital M, L capital M, N, L capital N, L capital N, M, N, then you will have L capital M, L capital N, and some references consider it a heterozygous individual. So if you are MM, the antigen on your red blood corpuscle is M. Here, the two antigens are present and they are equally dominant, and therefore we consider it a case of codominance. So which of these three genotypes, that one, or that one, or that one, is an evidence of codominance? No, it is the MN that is an evidence of codominance. Imagine now that I don't know what the genotype of my blood is, so I'm going to use anti-sera. Anti-sera are sera, or sera is the plural of serum, sera containing antibodies, containing specific antibodies. So what are anti-sera? They are sera, and those sera are containing a specific antibodies, okay? And I'll take a blood drop, a peripheral blood drop from my finger, and I divide it into two, and then spread it on a slide, and then add anti-M to one drop and anti-N to one drop. If I am of blood group M, then see here, there is even distribution on the slide, but here there is clumping or agglutination. This clumping or agglutination means that an antigen-antibody reaction has occurred, and this is the result of the antigen-antibody reaction. The antibody is anti-M, so the antigen should be M, so I know that I am M. If I am N, then if I add anti-serum N, clumping will occur. Clumping will occur. But if I add anti-M serum, clumping will not occur because I don't have the M antigen. I'll pause for a second. Okay, hello again. We will resume and see what the next slide is talking about. Now we have a case of multiple alleles. So the fourth non-Mendelian case is a condition of multiple alleles, and we have just said that the inheritance of the ABO blood system alleles is an example of multiple alleles. Of course, since most of the genes in the populations, in the human population, exist in two allelic forms, then they are Mendelian. But sometimes a few kinds of inheritance, like inheritance of the blood group, for example, you have more than one pair of alleles that controls one phenotype. So, For example, in inheritance of ABO blood system, we have 3 alleles, A, B, and O, and they control one phenotype, which is the blood group, and that's why we call them multiple alleles. But at the end of the day, you receive only 2 of those alleles on your 2 loci, and they are represented as 1 or 2 antigens depending upon complete dominance or lack of dominance. So what are the possible genotypes in such a case? For example, we have 3 alleles, but we have only 2 loci. So I might receive A capital, or A capital O, or B capital, or B capital O, or I might receive A capital, B capital, O, O. So these are, we have 3 alleles, 3 different alleles. We have only 2 loci, and we are studying all the possibilities that those 3 alleles are distributed on the 2 loci. Now, if you have this genotype, then your phenotype will be A. Your red blood corpuscle will have antigen A. Similarly, if you have this genotype, because A is completely dominant over O, you will still have the same genotype. If you have the genotype B capital, because you are pure, you have 2 similar alleles, then there is no problem, and your red blood corpuscle will carry the B allele. And similarly, if you are BO, you will also be, your phenotype will also be B, because O is recessive to B. Here, of course, you will be O. And it is important to note that this allele, this recessive allele O, is not represented by an antigen. So your red blood corpuscle will not have any antigen on the red blood corpuscle. But if your genotype is A, B, because A and B are co-dominant, then you will see both of them on the red blood corpuscle. So actually, blood grouping is an example of multiple alleles. This is the first inheritance case. It is also an example of co-dominance, because A and B are equally dominant, and it is an example of complete dominance, because A is completely dominant to O, or O is recessive to A, O is recessive to B. So it is possible that in one inheritance case, you follow Mendel's rules. Some alleles follow Mendel's laws, like A in relation to O and B in relation to O, or they might not follow Mendel's rules, like A in relation to B. So here you can see in that table, you can see that in group A, you have antigen A on your red blood corpuscles, but of course in your plasma, you will have anti-B. In group B, you have antigen B on your red cell membrane, but of course in your plasma, you just have anti-A. If you are AB, then of course you cannot have any kind of antibody in your plasma. And if you are group O, then you will have both, you can have both anti-A and anti-B in your plasma. Concerning the antigens, as we have just said, group A has the RBCs of group A have antigen A, the RBCs of group B have antigen B, the RBCs of group AB have both antigens A and B, and the RBCs of group O don't have any antigens on the red cell membrane, and that's what allowed them to have anti-A and anti-B. As we said before, that blood transfusions should be compatible blood transfusions, and that we should test first. Of course, we do the blood testing, and we know, for example, that the donor is an O group, for example, and the recipient is an A group. However, we must do cross-matching again for further emphasis that the two blood groups are compatible with each other. So compatible blood transfusion means that the donor's plasma, the recipient's plasma, the recipient's plasma, what is important is the recipient's plasma. Why? Because so here the recipient plasma has antibodies, of course, or does not. But if it has antibodies, so it is very important that we focus on the antibodies in the recipient plasma and the antigens on the donor RBC. So here we have an antigen on the donor RBCs. Why don't we care about the plasma of the donor and the RBCs of the recipient? Because the plasma of the donor is a small volume and it would be diluted and have no effects when received by the recipient. So here, for example, if you have on the donor's RBC, if you have on the, let me choose red, if you have on the donor's RBC antigen A, so of course you cannot have anti-A here. You cannot be of blood B. So blood group B cannot receive from A. And as you know from our study in biochemistry that group O, since group O does not have any antigen on the red blood corpuscles, then group O can be considered a universal donor because however the antibodies, whatever the antibodies in the plasma of the recipient here are, group O doesn't care about this because it does not have antigens. So group O is a universal donor. Similarly, group B is considered, group B is considered a universal recipient. Why? Because group B does not have any antibodies from the start, from the very beginning. It does not have any antibodies in the plasma, so it can receive whatever type of blood with whatever types of antigens on the red blood cell membrane. Of course, this is another way how we can represent the phenotypes by genotypes. But actually, we have two, as you can see, we have 6 genotypes, but for example, this genotype and that one are both group A. That genotype and that genotype is alone, which is AB, and then we have that one, which both together give B. So actually we have 6 different genotypes, but we have 4 different phenotypes. You have A that is repeated, and you have B that is repeated. That's why we have 4 phenotypes only, or 4 blood groups only, A, B, AB, and O. Although we have 6 genotypes, this comes from where? This comes from the fact that A is completely dominant over O, and B is completely dominant over O, okay? Okay, thank you so much, and then you will be receiving very soon part 5 of heredity.